insects behavior, and she was one of the stars of the class. So I didn't hear from Arden until February of 2020, when you contacted me about the possibility of working in the lab. And I don't know if you remember that it took a little while to get things going, and by the time we actually formalized it, we were April 2020, and so it was a digital joining of the lab. And as with Lucas, because we couldn't do anything in the lab, we brainstormed about what she could do, and so we had this idea of Arden with her graphical skills doing a graphic novel, telling a story of one of the research projects in the lab. And here we are with a test print of it. And it's one of the most exciting things I've had the pleasure to participate in my career, and it's been fantastic doing it with you. It's really been a joy. So the plan is for this to finish this summer, to be finished this summer. So in fall of 2002, Arden began a master's degree in my lab. She proposed to do something with insects and behavior, which was way outside of my comfort zone. But because of some excellent committee members who, along with Arden, brought me up to speed, it's taken me to realms I never dreamed I could go, and it's taught me a great deal. Arden, it's been a delight having you in the lab. And with some sadness, we get to this point of a defense and a departure. And so I will pass it on to you to tell us about your project, but let's wait for David.
We get to look at the book later.
Yeah,
Yeah.
you can look at it now.
Okay.
Are you going to submit it to a publisher, Arden?
So that's a little complicated.
So the plan A was to have it published. Oh, we'll tell Sarah. Okay.
Okay, well, something's going on here, yeah.
All right, well, thank you so much, David, for that introduction. I appreciate it. It's been a great few years. So, hello, everyone. Thank you for coming this morning. I'm going to be presenting the results of my master's thesis research that I've been working on for the last 3 years. So this is slime and seed, exploratory and foraging behavior in forest-dwelling carabids and cicadelids. So just a sort of a roadmap of everything that we're going to be talking about today. First, I'm going to go over some background in animal behavior, especially animal personality. Then go over some details of our study group. Then get into some of the methods and materials, including the specific species that I looked at in this project, as well as the different experimental design for the different trials that I used with the beetles. Then get into some results in terms of the beetle exploratory behavior, foraging behavior, and how those two behaviors come together. And then finishing off with some last conclusions and next steps. So let's start off talking about animal behavior, specifically with regards to animal personality. So animal behavior is something that drives almost every interaction that an animal has with its environment and the other animals, organisms that live within it. So animal behavior contributes to mating success, to procuring resources and foraging, and to responding to threats in the environment. And in a lot of ecological modeling, it's a lot easier to think of behavior as this cohesive thing across a species, as where it's sort of this unit with consistency across the population. However, thinking about animals in this way can sort of flatten a lot of the variation of behavior that can occur between individuals, because these populations are made up of individuals who often exhibit a lot of variation in different behaviors. So variability between individual behavior in a population may even improve survivorship on a population level, because if you're living in this changing environment and every individual in the population acts in exactly the same way, this may not be beneficial depending on what's going on in your environment. So one particular aspect of animal behavior, especially with regards to individual personality, that is a growing area of research is this idea of animal personality. basically, animal personality means that individuals in a population exhibit correlated clusters of independent behaviors, which are consistent and repeatable across time and vary between individuals. So if populations exhibit these suites of correlated behaviors that have this individual consistency across different contexts, that is when we can say that an animal exhibits a behavioral syndrome. Now, this is something that has been observed in a wide variety of animal species, a lot of mammals, other vertebrates, but it is also growing subject of research in invertebrates, including insects. So animal personality is typically measured across 5 main axes, which includes shyness and boldness, exploration and avoidance, activity, sociability, and aggressiveness. Then they can vary along each of these axes depending on their behaviors. So the axis of animal personality that we are most concerned with in this study is going to be this axis of exploration and avoidance. So exploratory behavior is a pretty critical axis of behavior when you're looking at how an animal engages directly with its environment. So exploratory behavior is linked to success in foraging, in finding mates, in access to other resources. However, increased exploration can also be costly because it is energetically expensive and it increases the risk of encountering dangers in the environment, such as predators. So in the lab, we typically measure exploratory behavior using an assay called an open field test, which is an experimental design that was first used by Calvin Hall in 1934. And he used this design to measure defecation of rats as a measure of timidity in the open field. So basically, the idea behind this experimental design is to measure the behavior of an animal in an open, novel environment. And oftentimes, especially in small species that are vulnerable to being eaten by other animals, the risk of predators is heightened in the open field, which leads to a lot of wall and corner seeking behavior in these animals. So now that we've got a little bit of background on behavior and personality, let's talk about our study group for this project. So caravid beetles, family Carabidae, are a large and diverse family of beetles with over 40,000 species found worldwide. The majority of these insects are carnivorous, and a lot of them are generalist predators that eat lots of different little invertebrates and other insects. However, some of them are omnivores or exclusively seed feeders, and some are specialist predators of specific kinds of invertebrate prey. There are some that prey on specific millipedes, or springtails, or slugs, or snails. So one group of specialists that we are particularly concerned with for this project is Troutcytokine, which are these snail-eating specialists. They have specialized morphology that allows them to eat snails in their shells and have these elongated heads and mouth parts, which I'll show some pictures of a little bit later. So despite this family being so large and diverse and widespread, there's not been a huge amount of study into their individual behaviors and specifically their personalities. So personality syndromes have been studied in 2 species of ground beetle. This is Carabus convexus and Vibrio revocalis. And then Vibrio revocalis is the one shown here on the right. So Vibria is particularly relevant to this study because both of these species are natively found in Europe, but Vibrio revocalis has been introduced in the Willamette Valley in the last couple of decades and has become extremely common in this region. So in a lot of studies on exploratory behavior in carabids, they will use exploratory behavior as a potential correlate to other behaviors or other environmental factors such as urbanization or associative learning. However, some of the results of these correlations seem a bit mixed or context dependent. So a study has found that urbanization is a factor that's linked with increased exploratory behavior in these beetles, but a lot of times it appears to be sex and season specific, and it's not like a universal pattern that occurs across the entire population. There was also a study that looked at exploration in Vibrio revocalis as correlated with the ability to perform associative learning tasks. And while the the beetles were able to perform these tasks and they varied in their exploratory behavior, there wasn't any correlation found between exploration and the ability to learn. So as I mentioned, there are both specialist and generalist species of caravid, especially ones that are specialized to prey upon terrestrial gastropods. And so in the lab, both specialist and generalist species of caravid have shown evidence that they can orient to gastropod mucus cues. However, not all studies have shown this. There have been some where even the specialist species haven't been able to reliably orient to gastropod mucus. It can be somewhat variable. Some specialists also show preference in terms of snail size and species that they prefer to eat. So the thing about generalist predators as predators specifically of terrestrial gastropods is that a lot of these gastropods have specialized morphology to prevent being eaten by bigger animals. And then generalist caravids typically lack a lot of specialized morphology in order to combat these challenges. So things like defensive mucus secretions from slugs or snail shells, these things can be difficult to overcome for generalist predators. However, a lot of them still do orient to mucus in the lab. And so it's thought that orientation to mucus could possibly be a strategy for finding other prey aggregations, even if slugs and snails are not a primary prey item of these generalist species. So mucus studies in caravids typically use this Deroceras reticulatum slugs, the gray field slug, which is a common agricultural pest in this part of the world. And so caravids have also been assessed as a potential biocontrol agent to combat these slugs. All right, so now we've got a little bit of background on our study group. Let's talk about the species that we used specifically in this project. So the beetles were collected from 3 localities here in Benton County: the Mary's Peak Campground, the OSU Cow Farm near Oak Creek, and the Lewisburg Saddle Old Growth Trail in McDonald Forest. So each of these sites contains at least a small riparian zone, which is an important habitat for a lot of the species. But one thing to note overall about these localities is that they were selected mainly to retain a diversity of beetle species and not necessarily due to the diversity of habitats in and of themselves. So we weren't necessarily expecting a strong result due to habitat in terms of the beetle's behavior. So the beetles were hand collected after dark throughout the spring and summer of 2024. So these are little nocturnal predators. So after dark, they come out and start running around the forest floor. And the spring and summer are a period of high activity in terms of their seasonal life cycle. So this is the phylogenetic relationships of the 9 species that were used in this project. So I'm just going to briefly go through some of the major attributes of each one. So these 2 species, Omus algerni and Omus dejani, are the only 2 species that are actually not caravids. They are tiger beetles, family Cicindelidae. They are generalist predators, and they have these very large mandibles that are characteristic of tiger beetles, and they tend to be a very bold and aggressive species in the field, in my experience. They are not afraid to use these structures. So next, moving on to the caravids in the study. So these 3 species are in that tribe Cyprinae that I talked about. They are the snail specialist species in the study. So this is Cyprus tuberculatus, Acknowledgments and Acknowledgments marginatus. They tend to prefer slightly more riparian and more secluded habitats. And then one thing that's particularly interesting about this guy in the middle, Acknowledgments, is that they exhibit this pretty unique tree climbing behavior. So in the field at night, they will climb many, many meters up into trees, which is pretty unique for ground beetles. As you may guess by the name, a lot of them don't climb very much and spend a lot of time running around on the ground. So this is some of those specialized morphology that I was talking about. They have these elongated heads and mouth part. They can stick into the shell of the snail in order to eat the snail without crushing the shell. They also have these very large exaggerated palps, which they use as part of their sensory system to navigate their environment. Next, we have Mebria rivercalis, which I also mentioned in the background. This is a species native to Europe, has become a very common introduced species here in the Willamette Valley, as well as the region. So this is also a generalist predator. and is slightly more tolerant of colder temperatures and more disturbed habitat compared to some of the native species. It has also been shown in previous research to exhibit individual variation in personality. So Zacotis matthewsi is a very striking beetle. It is pretty unique. It's the only species in its genus, and it is another generalist predator that tends to prefer more secluded habitat in forests. And then lastly, we have the two Pterostys species. So these are both within the subgenus Hyperpes. They're both generalist predators. They are both found in forests, but Pterostys algidus is a little more tolerant of open habitat than Pterostys lambda. So this is the lineup of all 9 species. Kind of see the diversity of size here. And then this is the breakdown of samples across all of the species from each of the 3 localities. So most of the species were collected mainly from one locality, a couple of them were found in 2. And the overall species samples ranged from an N of 6 to an N of 26, but most of them were over 15 to 20. Then the total number of species, or total number of individual beetles included in the study was 173 across the 9 species. So after collection, the beetles were housed individually, acclimated for 1 week, and then fasted for the duration of the experimental period. So lastly, we have our gastropod. So this is Vespericilla columbiana, which is the Northwest Hesperian. It is observed to be a prey of Sciathinotus marginatus in the field. It is a relatively common snail species that has a similar distribution to the study beetles, so it is a potentially a prey item of the other species in the study as well. All right, so after we got all these beetles, what did I do with them? So in order to investigate beetle exploratory behavior, there were a few questions that I was hoping to explore. The first being, how does exploratory behavior differ between species as well as other demographic factors? And do individuals exhibit consistent variation in their exploratory behavior over time? So in order to investigate these questions, I used a variation of the open field test. So I used this open arena. It was a 20 by 10 inch tank, basically. And so the beetle was placed into the center of the tank and then allowed to run around. So then I filmed the beetles with a GoPro for the duration of the trial. Then I can upload these videos into an RStudio package called Path Tracker that generates these plots with paths and heat maps showing where the beetle went and where they spent the most time. Then I can composite all of the plots for all individuals of a species and create these final composited plots showing all individuals of the species overlaid. And I will return to these visualizations later. But this is what the experimental design actually looks like. So this is the tank and the GoPro. And so all the trials were performed under red light because beetles are nocturnal. And so this is in the interest of trying to replicate more natural behavior during their natural period of peak activity. One thing that was slightly changed from this image, this is more of a preliminary version of the design, but I did end up covering the walls of the arena as well, just in case they were able to see at all under the red light, to make sure they couldn't see out of the arena during the trials. So the exploratory trials lasted for 120 seconds and were replicated 3 times over the week-long experimental period on alternating days. So in order to quantify exploratory behavior, I took several different measurements, including latency to initiation, which is the number of seconds until the first second of sustained movement, basically how long did it take them to start running around. The distance that they moved throughout the trial, the number of cells that they crossed. So I digitally imposed this 5 by 10 grid onto the arena during tracking, and then counted the number of cells that the beetle interacted with throughout the trial, basically as a measurement of how much of the arena they explored. The time in motion throughout the trial, and then time in the center region, which was defined as more than 2.5 centimeters away from the wall, which is usually a measure of boldness because timidity is associated with this wall-seeking behavior because the small animals tend to feel vulnerable to predators in the middle of the open field. And then lastly, I combined several of these measures into an overall exploratory score for each beetle in each trial, which is on a scale of 0 to 1. All right, so now let's take a look at how I tested their foraging behavior. So I also had a few questions about the foraging behavior of the beetles, including, do beetles orient to a snail prey cue over an alternative treatment or a control? Do beetles respond differently to foraging treatments based on species and other demographic factors? And do individuals exhibit consistent variation in foraging behavior over time? So in order to test these questions, I used a slightly different adaptation of the open field test. So this one was in a circular arena that was 15 centimeters in diameter that had 3 zones that we created in the arena. So one of them was this mucus treatment from our Vespericilla columbiana snails. Another was a Vaseline treatment that was just manually applied to this zone. And then the last was an empty control with no treatment applied. So this is how the mucus treatment was applied. So I got my helpful little Vespericilla columbiana to crawl around and put natural locomotor mucus onto this region of the arena. Usually took about 20 minutes until the zone was fully covered with an appropriate layer of mucus. Then, once the treatments were applied, the beetle was placed into the middle of the empty control zone, facing the center, and then was allowed to move around freely. So these trials lasted for 1 hour and were also repeated 3 times on alternating days to the exploratory trials during the week-long testing period. So in order to quantify foraging behavior, the measurements that I took were zone entries. So whenever the beetle crossed over these borders into a new zone, that it would have been entry to that zone. And I also looked at the proportion of time spent in each zone, specifically after their first entry into the mucus treatment zone, because a lot of beetles took a little while to start moving after the beginning of the trial. And so I didn't want it to be skewed toward too much time in the control zone because the beetle hadn't yet properly interact with the treatments. All right, and then one of my final questions was how these 2 things are related. So I asked, is there a correlation between beetle exploratory and foraging behavior based on variables in each of these tests? So let's get into what I found, starting with exploratory behavior. So you'll remember, this was my experimental design for exploratory behavior. I take these videos of the beetle movement in the arena, and then I can use Path Tracker to generate these tracking plots that have a path for the beetle as well as a heat map showing where they spent the most amount of time. Then I can digitally composite all of the plots for individuals of a species to create these overlaid plots that have heat maps and paths overlaid from all of the individuals in a species. And when I do that for each of the 9 species, it looks something like this. So right off the bat, a few things you might notice are this wall seam behavior that is very common in most species, which is expected in small animals in the open field. But there's definitely some variation in movement where some species seem to be moving around a lot in the arena, resulting in a lot of these overlaid paths on top of one another, and then some that exhibit less movement in the arena overall. So let's get into some of the more quantitative measurements of exploration. So in order to test the effect of some of these fixed and random effects on variation in exploratory behavior, I created these generalized linear mixed models and then compared them through stepwise ANOVA comparison in order to test whether each factor was a significant predictor of the diversity that was observed in exploratory behavior. So I did that for each of the different measurements of exploration that I outlined in the methods. So as you can see, species is a good predictor of a lot of the variation that we're seeing in exploratory behavior in most of the measurements that I took. So it's not a good predictor of time in the center region because this is something that was very low almost universally. Most of the beetles did not like to spend time in the middle of the arena. So if you look down at the bottom here, individual ID was this random effect. So I did this in order to see if beetles were exhibiting consistency in behavior over time. And so it did come out to be a significant predictor of variation for several measures. And then this conditional R squared here, this is showing is the fit of the model with individual ID included as a random effect. And then this marginal R squared is the model fit without the individual ID. So the model fit is variable between these different measurements, but in all cases, including individual ID does improve the overall fit of the model, which seems to indicate that individuals are exhibiting a degree of consistent behavior. So let's get into some of the variation that I observed in terms of exploratory behavior by each species. So this is showing the distribution of exploratory scores for individuals separated by species. And there seems to be a good amount of variation going on here. So one thing that jumps out immediately is the differences in behavior between the three cyprines, the snail-eating beetles. So Cyprus tuberculatus and Scaphonotus marginatus have pretty low exploratory behavior compared to the other species in the study. However, Scaphonotus angusticalis had actually the highest mean exploratory behavior of any species, even though it's so closely related to Scaphonotus marginatus, which was not very exploratory at all. You can compare that to some of the other pairs of closely related species groups in this sample. So we have the two omus species up here, and then the two Terosticus species down here. And each of these pairs, they kind of have like middle to high exploratory behavior, I would say. And they seem to exhibit a lot of consistency within each of the two genera. And then lastly, I want to call your attention to these three species. So I already mentioned Cyprus and Scaphonotus marginatus as two of the least exploratory based on these findings. And then one of the other less exploratory species was Beetles, although it's a little bit higher, it's kind of like mid to low range of exploratory scores, I would say. But you can compare these three species to these three composited plot outputs. So I think it's interesting that you can see some of this corroboration between the more qualitative measurements in terms of exploratory score. I think this maps pretty well onto the more qualitative measures of what these plots look like and our visual perception of the beetle exploratory behavior. Then the last thing that I want to call attention to from these models is this slight effect due to trial that was observed in time in the center region and in overall exploratory score. So this is the result of trial in both of these measurements. So we can see that time in the center region over each of the three trials was found to increase somewhat into the second trial. And then exploratory score was found to decrease slightly by the third trial. Now, this isn't a huge decrease. It's only like by a few seconds on average, and then by like less than a 0.1 difference in exploratory score. But it's possible that this could be the beginning of evidence for habituation to testing conditions, where maybe the beetles felt more comfortable in the arena after prior exposure, so they were willing to spend more time in the center region. Coupled with exploratory score, maybe they're less likely to explore an area as much if they're familiar with it already. But I think it would need more research over a longer period of time and more trials in order to really see if there's going to be a strong effect there. So collection locality was not found to be a major factor driving exploratory variation, which, as I said, is pretty much to be expected. There's not a huge amount of variation between the three habitats, although the Oak Creek habitat is more disturbed and high traffic than the others. It still has like small areas that are forested and have a creek and are not super high traffic. So maybe on a microhabitat level, there's just not enough variation to really see a big difference here. But there's also something to be said for the fact that interspecific differentiation within an ecosystem may be beneficial, where if all species that are in the same habitat are acting in similar ways, that may put them in greater competition with one another for resources. So this is something that I think we need more study over a wider variety of different habitats and biomes to really investigate. And then lastly, the behavior of the introduced species, behavior of polyps, was pretty consistent with native species, even a little bit low. So it doesn't seem based on these findings that exploratory behavior of this introduced species seems to have a significant effect on invasive success, but this could also be something that is season specific or habitat dependent. Maybe there are populations of Neobrevibrocollis collis that are more exploratory than others that weren't captured by this study. It's difficult to say. All right, so now let's talk about their foraging behavior. So I used a similar technique for measuring testing measures in the foraging trials. So using this generalized linear mixed modeling to test the fixed effects and then the random effect of individual ID. So species was shown to be a good predictor, again, of variation in the number of entries to different zones, as well as proportion of time spent in different zones. Diet was also found as well, but not necessarily in the way you might assume, so I'll talk about that a little more in a moment. And lastly, individual ID was also shown to explain a lot of the variation in the number of entries to different zones. So beetles were showing consistency in their movement patterns in the foraging trial. So this is the breakdown of entries to each treatment zone. There was not a lot of variation in entries between the different zones. In fact, no beetle showed significantly different entries to any given zone. However, there does seem to be a lot of variation in terms of the overall entries. Basically, this is showing how much they were moving around the arena and crossing over into different zones. And so this may be another pattern that has some sort of phylogenetic link. You can see that the Oma species are pretty similar, the Girosticus species are pretty similar, and even the three cyprines seem a lot more consistent in this behavior than they did in their exploratory behavior in the exploratory trial. Right, so now for the fun stuff. So this graph is showing the relative proportion of time that each species spent in each zone on average. And this shows a lot of variation. So first thing I want to point out is these two species. So Scaphnotus marginatus, one of the snail specialists, and Zacodus matthewsi, one of the generalist carabids. These were the two species that spent a significantly greater proportion of time in the mucus treatment zone compared to the control. And then these four species, the four generalist carabids, all spent significantly more time in the Vaseline zone compared to the control. However, this species, Omastejani, one of the tiger beetles, was the only beetle to spend a significantly decreased proportion of time in a treatment zone compared to the control. It actually spent less time in the Vaseline zone. Now, this is a little strange and interesting, and there's a lot of factors that could potentially be driving this behavior. One of which being an effect of novelty. So it's conflict versus neophilia versus neophobia, where neophilia is where animals are attracted to novel stimuli in the environment, but neophobia, they're more reluctant about that stimuli. And so neophobia can allow for access to novel resources, but it can also be risky if you don't know what you're investigating. It could also be that beetles are responding to Vaseline as more of a tactile foraging cue rather than an olfactory one, because Vaseline is more viscous than the Vespericula columbiana mucus that is used in the mucus treatment. And it could also be that Vespericula columbiana aggregations are not preferred or beneficial to generalists in the way that they might be for a snail that is specialized to prey on that species. Difficult to say. Lastly, there could be some effect due to chemical signaling, which is extremely important in these species. So Vaseline is basically made up of a pile of different hydrocarbons and alkydes, and a lot of hydrocarbons are found in things like the insect epicuticle and in beetle contact pheromones and defensive secretions. So these things can vary a lot and are often very specific to different species. So it's difficult to say for sure what that possible effect may be, but it is possible there is some sort of overlap between chemicals found in Vaseline and those found in different chemical signaling factors in either the species in this study or in other species that they share a habitat with, which may cause a differential response depending on species. It could also be that different species are responding to each of these factors differently, and that they're not all following the same universal patterns, and they may have different priorities in terms of their response to Vaseline. So lastly, I want to look again at this effect due to zone, which was observed in the foraging trial, just like it was in the exploratory trial. So again, it's not a huge difference from each zone, but it was a slightly significant decrease going toward the third trial in overall zone entries, which may indicate less movement overall in the arena, which again could be linked to possible habituation to testing conditions. However, the proportion of zone or proportion of time spent in each zone was not found to vary really at all. It's actually remarkably consistent between the trials. So now let's take a look at how these behaviors come together. So in order to look for a potential link between exploratory behavior and foraging behavior, I took the number of zone entries and mapped it to a linear model with exploratory score, trying to see if the number of zone entries would increase as the exploratory score increases, more exploratory individuals having more encounters with the foraging cues. And I found a slight positive relationship there. You can see in this solid blue line, this is the trend that includes all individuals across all of the species. However, when you break the model down into individual species, the pattern sometimes follows the overall trend, but also becomes a lot more variable in different species, some of them even exhibiting a negative trend. So I wanted to see whether this was the result of sample size having an effect on these correlations. So what I did was resample the entire multi-species data set 1,000 times at a subsample that correlated with the sample size of each of the individual species. And so basically, these distributions show the slopes from those 1,000 models that correspond with the subsample replicants. And so this dotted line in the middle is the actual overall slope of the full data set. And so the smaller sample sizes don't do as well at estimating this slope, but they all do kind of center around the true slope, even though it's small subsamples. And then in red, you can see these distributions contrasted with the actual observed slopes of the models for the real data for the individual species. And so for some of them, they do seem to vary a lot from the observed relationship between zone entries and exploratory score overall. So it could be that this is a pattern that is variable between species, or it may require more testing in order to really investigate the role of exploration in foraging behavior. So let's just sort of summarize a lot of what I talked about and get into some conclusions and next steps. So you'll remember one of my questions about exploratory behavior was, how does exploratory behavior differ between species and other demographic factors? And there was definitely a lot of variation, primarily as a result of species that we observed. So some of these patterns may share a phylogenetic link, especially looking at ones like the Ulma species and the Terrastia species who exhibit quite similar exploratory behavior, but not all of them necessarily share this pattern, particularly the Scaphenotis, who have very different exploratory behavior. The phylogeny is not something that was included as a statistical control in these results, but it's something that we've been working on and are hoping to include going forward, thinking about publication of the project. So next, I asked whether individuals exhibit consistent variation in exploratory behavior over time. And it does appear that they do. It seems like individual variation was a significant factor in explaining the variation that was observed in terms of exploratory behavior. However, there was also a potential effect of habituation to testing conditions, which has some interesting implications for the design of these kinds of studies. A lot of similar studies to this just stop at two replicates of the trial in order to establish individual repeatability over time. So it may be that doing more than 2 or 3 might not be necessary, or maybe even detrimental if you're just trying to establish consistency of behavior. But I think it would be interesting to look at a lot of replicates over a longer period of time to see if the beetle's behavior changed more dramatically over a longer period of habituation. So next, I wondered about foraging behavior. Do beetles orient to a snail prey cue over an alternative treatment or control? And I did not find this strongly to be the case. Only 2 species did this, one of them being one of the snail specialist species. However, even the other 2 snail specialists, the Cyprus tuberculatus and Scaphenotis angusticalis, did not appear to prefer the mucus treatment to a control. Now, there's so many reasons this could be the case, including prey preferences. Maybe they don't like the kind of snail that I used. Maybe they have variation in foraging behavior where their hunting behavior is different, such that it wouldn't really map to spending more time in a mucus treatment zone in this kind of test. It's also worth pointing out that the behavior between these 3 species, the cyprines, seems to show a lot more variation within the clade than some of the other closely related species in the study. This aligns with my observations of their behavior in the field. As I mentioned, Acknowledgments is both very exploratory in testing and is also very active and climbs high into the trees in the field as well, which is pretty unique behavior. So I think that further investigation of the exploration, the diet preferences, and foraging habits in more species across this clade would be really valuable in starting to piece together these unique aspects driving the variation in their behavior. So my next question about foraging behavior was, do beetles respond differently to foraging treatments based on species and other demographic factors? And there was definitely some interesting patterns that seem to result from the Vaseline treatment. So all 4 of the gemmerless corallid species preferred the Vaseline treatment. There was no positive or negative preference found in the 3 snail specialists. And there was a decreased preference in 1 of the 2 tiger beetles. Now, I discussed some reasons that this may be, maybe an effect due to novelty. It could be something related to chemical signaling. It could be related to foraging, that they're just responding to cues differently. And I think it's probably likely that different species are responding to things for different reasons, especially with regards to this Omistijani, which was the only one to display a negative preference toward the treatment. So my last question about foraging behavior was whether individuals exhibit consistent variation in foraging behavior over time. And this was also found to be the case. So individuals exhibited consistency in their zone entries, which could map onto encounters with foraging cues. There was also in this assay potential evidence for habituation to testing conditions, which has a lot of similar implications to the findings from the exploratory trial. So my final question about if there was a correlation between beetle exploratory and foraging behavior appeared to be a little bit mixed. So overall, more exploratory individuals did enter more zones in the foraging trial, but this pattern doesn't seem to hold for all species, or at least not reliably enough to see on a small sample size. So it could be that different species exhibit different foraging strategies, which could cause them to respond differently to testing, either because exploration serves different functions in different species, so maybe foraging is more or less strongly linked with exploration depending on species, or it could be that cues in the arena influence exploration. So perhaps some of them become more exploratory in the presence of foraging cues, where others do not. So in closing, exploratory and foraging behavior in the species in this study is extremely diverse and varied. And it's not always clear what is driving these patterns of variation. There's a lot that we don't know about these little beetles and what they do. There's a lot of further study of these cryptic species that is really needed in order to understand their behavior and how it connects to their unique aspects of their physiology, their life history, and how they interact within their environment. So there were a lot of people who really contributed to making this project possible. So I have to thank my advisor, Dave Madison, who has been a great mentor to me for many years in this and other projects. And so I really appreciate his guidance and support, and you have given me opportunities that I could have imagined on my own. So it's been really special. And it's been great learning about all these little beetles too. I really enjoyed it. And thank you as well to everyone else in the Madison Lab, both current and former, who've just been a great group of people to work with, and I really enjoyed my time here over the last few years. I need to thank as well the other members of my thesis committee, Jamie Cornelius, David Cucci, and Julia Jones, who have really gone above and beyond in helping me develop this project every step of the way. Definitely could not have done it without your feedback and support. And I also just want to thank the rest of the IB department. I have been in this department since I was an undergrad in 2017. So it's a little wild to think that it's been that long and that it's going to be coming to a close. So it's been a great group to be part of for this long. I also need to thank everyone who came out to collect beetles with me last summer, which was a bit arduous, but it was really fun and could not have done it without all of your help as well. Of course, I also need to thank my family and friends for their support and enthusiasm over this entire process. They are always so excited and interested in what I'm doing, even if they don't always know or quite understand why I'm doing the things I'm doing. But I could not have done it without their love and support, so thank you. All right, and then this is my final plug. So I was a part of the Pratt Student Fellowship this term, which is basically an art science initiative that's combining aspects of different STEM disciplines with different aspects of the arts. So David was my mentor, as well as Andy Myers from the art department. And so I worked with them to create these paintings of a few different species that were included in my project. So this is Omastajani, Scaphosaurus marginatus, and Zacotus matthewsi. So these paintings are still up on view in the Prax building on campus. So I would encourage you all to go check them out, along with all of the other amazing artwork that people created as part of the fellowship. But with that, I will take any questions.
Yeah, I was just curious, did you do any initial trials on the size of the arena and if that mattered in their behavior? And then also, second question is, for the initiation thing, I was wondering if you put them into the exact same arena that they perhaps already went in, so maybe they smell, like their own residue. I don't know how bugs work, but you know what I mean? Like,
Yeah.
or was it like a completely new one that, like you cleaned and then it was novel again?
Yeah, so I did clean the arenas after each trial, and then the beetles were also tested, they were never tested more than once on the same day. So I don't think that it's a result of recognizing their own smell necessarily. But yeah, as for the size of the arena, I did a little bit of experimentation. I did a lot of preliminary testing, in a previous year. And I mostly experimented with the size of the foraging assay. But one thing I've noticed is that the beetles' behavior, at least in terms of their movement patterns, tends to be very consistent no matter the size of the arena. I've also had beetles escape from the arena and get out into the testing room, and they basically behave exactly the same, where they just run, find a wall, and then start running laps the entire time. So it was also difficult because I had a pretty big variation in size of the species that I was looking at. So I couldn't go too small, but if it was too big, then that might not be good for the smaller species. So I tried to find a balance there.
So since rapid behavior, especially in the ones that you were looking at, is the study of it is kind of relatively new. Were there any sort of roadblocks or hiccups in developing the experiment or collecting data that you weren't really expecting with sort of their behavior, and how did you sort of work around that?
Yeah, so there was definitely a lot of that. Like I said, I did a lot of preliminary tests in the last couple of years before running my actual assays, just to figure out how to make an arena that they would behave normally in, different substrates really affect their movement, things like that. Also creating something that I could reliably film that was also contained, that they weren't able to escape from. I had that problem as well. So there were definitely designing the experiments in a way that made sense for species was pretty difficult without having a lot to go off of. Also just the fact that all of at least the personality studies have been done on species that are native to Europe. So I really didn't have much context for any of these species specifically, besides maybe Rhodopolis, because it's the introduced species. So that was fun in a lot of ways, but also a little daunting, just that there's not a lot of context for any of these species in terms of their personality variation.
Thank you.
Was there any correlation that you could kind of tell, just qualitatively speaking, between walking speed and their behaviors at all? Or did that not seem to affect things at all?
Yeah, I didn't notice a big effect due to walking speed. I would say just based on my qualitative observation, they all are pretty much either running or sitting still. I think the problem with run speed is that's gonna vary a lot based on size. And so I would have had to do a lot more complicated, like compensating for like leg length and things like that, which ended up just being beyond the scope of the project. But I think that would definitely be something interesting to look at. Size of the beetle and movement, like physical movement constraints and how that affects their.
Hey. Oh, that's, all right.